Okay, so this then is asking you to label two parts of the digestive system. This is your gallbladder by here, and this is your liver. Okay, the liver secretes a substance called bile. This helps the lipase digest fats into fatty acids and glycerol. State the function of Z and shown in the diagram. This is the bile duct. Okay, so the bile moves from the gallbladder into your bile duct. Okay, and the idea behind this is it moves bile into the small intestine. I just realised there's a slight mistake here. This is the stomach. Okay. Describe the effect pH on the rate of digestion of the lipase. Lipase works best in alkaline conditions, so you can see there's more digestion in these two than there is in these two. Acidic is the worst. Okay, there's a lot more digestion in these two. Explain the effect of bile on the rate of digestion. Bile massively increases it. So both of these are alkaline. You can see it's near enough another 50% increase, okay, on the rate of digestion. Again, a nice little question. There's a lot of kind of similar questions here. Describe the organ that secretes bile. That's the liver, and you have to label it. So there's one mark for saying liver and another mark for labeling it. And then the same then for storing bile is gallbladder, and you have to label that on the diagram. There's then a lot of information here on a table about what happens to water, oil and bile, and water, oil, bile and lipase. Okay, you can see here that water and oil have no effect on digestion, nothing digests. The same for water, oil and bile. It's only when you've got the lipase as well as the bile does the fats break down into fatty acids and glycerol. Okay, that's only when it would happen. Again, there was a little of an error in terms of where the line was. The liver is this one here. Okay, that is the stomach. Explain, bile plays a very important role in digestion of lipase, but from table 2 shows not directly involved. Okay, describe the role of bile. Bile acts as an emulsifier, provides a larger surface area for lipase to act. Digestion happens quicker. The production of fatty acids in tube 3 did not increase after 20 minutes. There's just a reason for this. No more fats or lipids to break down. Apart from fatty acids, name the other product. It's always fatty acids and glycerol. So a couple of quite easy recall questions. They even gave you the answers at the top. So protease, protein is amino acids. Lipids is fatty acids and glycerol. Then you kind of had to match up the enzymes, A, to which of these substrates it would break down. So A would be 3 because 3 would fit in the active site. B then would be 2 because it fit in the active site. D 1, and then C 4 because they fit in the active site. Then we're looking at describing a graph. So 3 marks for describing what you kind of see happen in this graph. From the graph, describe the effect of temperature on the rate of reaction between 10 and 50. So the temperature increases until about 35 degrees. After 35 degrees, enzymes will denature and the rate of reaction slows down. Use your answer in part B to explain why denatured enzymes can no longer work. Active site changes shape, substrate cannot bind anymore. Same kind of question that we've done before previously. These bind in to the active site. It's called lock and key. Explain what would happen if you boil them. Active site denatures, so substrate can no longer bind. Okay, remember it all changes shape, so these will no longer fit in there properly. Another label in the digestive system. What does it say at the top here? So, what is broken down into what? So, proteins are always broken down into amino acids. Fats, we spoke about this a lot. Fatty acids and glycerol. Starch is then glucose. Why is it necessary for these large molecules to be broken down? Smaller molecules can be absorbed so that we've got nutrients for energy, stuff like that. Okay. Function of carbohydrates, they provide you with energy. Label the diagrams. So you've got F here, you've got C, which is the small, oh, I've done that wrong. That is the small intestine. G is the large intestine. Okay, 2 letters where protein is digested, that would be C and E, so in the stomach and in the small intestine. Then looking at food tests, glucose is Benedict's and protein tests for your burette. Really nice easy question here then. Where is bile stored? In the gallbladder. Where is it made? In the liver. Really nice question, 2 marks there. Use the information in the graph. Describe the effect of bile on the rate of digestion. the use of bile increases the percentage of oil digested. Bile increases the surface of the fats, so there is more room for digestion to occur. It is an emulsifier. Again, what is diffusion? Movement of particles from a high concentration to a low concentration. Okay. A little question then about another viscid and tubing prac. So this could come up in any part of the cells or digestion. What is the function of the viscid and tubing? Is to What is the function of washing it into the tube stream of water for 15 seconds? Remove any unwanted substances. So this question offers a lot of marks, okay? So the first thing you've got is students have a sample of water, asked to sample distilled water in a beaker for the presence of both glucose and protein at the start of the experiment and every minute for 4 minutes. David decided to photograph his results on his smartphone and the photographs obtained show below, okay? And they even went as far as to tell you that Benedict's tests for glucose, Burette tests for protein. Explain David's results at 4 minutes for the contents of the beakers of both, okay? So you can already see the colour change, okay? So Benedict goes from blue to orange in the presence of glucose, okay? As you can see, the concentration gets a lot higher, so that means that a lot of glucose is being produced. So more glucose is being produced, okay, as the 4 minutes carries on, okay? Then this one, Burette, contains no protein, okay? What it should do, if you've got Burette, it should go from blue to purple, but there's no colour change. So then that means that there is no protein present, okay? Again, a nice easy question, labelling parts of the digestive system. You've got your liver by here, you've got your stomach, and you've got your pancreas. Starch is broken down to glucose by carbohydrates. Fats are broken down by lipase into fatty acids and glycerol. Test for protein is Burette solution. Again, another question looking at different parts of the digestive system. So label X on the digestive system. We're looking at the esophagus having come from your mouth into your stomach. It shows structure X. Part A of the ball for food enters structure X. Complete the diagram, okay? So what happens is that the muscles above it contract, the muscles below it react and relax, and that allows the food to move through, okay? Name the process, peristalsis. How muscle contractions occur, they contract and then relax, okay? State this name of structure Y, and it stores bile, that's the gallbladder. You can see that question comes up all the time. Describe how bile helps affect the digestion, increase the surface area to digest. These kind of questions come up all the time. Name the process in which food moves through the digestive system, peristalsis. State and explain what happens between pH3 and pH6, okay? So it increases quite quickly. pH3 is the smallest mass, where the smallest mass of food is digested, and then until about pH 5, where it levels off. So pH 5 is where it levels off. Optimum, so the mass of undigested food is 3. That's where you don't want any undigested food, so it would be 3. Name, so the organ is the pancreas. It's very likely to be protein because it works at such low pHs, so it works in the stomach. Again, another question on peristalsis. Another example of it, naming it for 1 mark and then explaining what happens. So contraction of the muscle above the food forces the food down, whereas this bit of the muscle relaxes so the food can move into the open space. And then really nice last question there. State which of the label parts in the above diagram absorbs digestive food, and that's the small intestine.